Hello, my name is Bradley Alicea. I'm from the Open Worm Foundation and Orthogonal Research and Education Laboratory, and I'm the head of the Diva Worm Group. And I'm going to talk to you about our work on digital diatoms, biophysics, and information processing. So at the right here, you can see some examples of the digital diatom, which we will be talking about today. So first, we start with a short introduction to diatoms. So this is a model organism we've been working with over the past several years. It's a very simple organism, and it has a lot of properties that lend itself to digitization. So we do this work in conjunction with some of the developmental work we do with C. elegans and other model organisms as well. So diatoms are algae. They're single-cell eukaryotes with plant-like characteristics. So they have a chlorophyll and a silica cell wall. So the cell wall is actually important because it keeps it rigid, and in fact, it can keep its rigidity in a number of shapes, as you can see at the left. Picture at the left, you have circular phenotypes, you have rod-shaped phenotypes, you have other types of elongated phenotypes, and you even have things like stars and other types of shapes. So it's a very fascinating array of shapes. We can look down at the bottom and see that there are these rod-shaped, flake-shaped, and 3D-shaped phenotypes. And they're all about 10 to 80 microns in size, which is not very large. So they're very small organisms. Sometimes they live solitary, and sometimes they live in colonies. And we'll be talking about a colonial diatom called Basilaria. That's the genus name. We'll be talking about that today. So up at the top right is an example of a raphid diatom. And this is an example of the Basilaria diatom, a single cell of that colony. And as you can see, it's an elongated shape. It has different features on the cell. So it has a silicate wall, so it's rigid. It has a frustrule, a hypovalve girdle bands, so along the edges it has structure. And then it has raphe, which are in the middle, and then an epivalve, which are on the sides. And so you can see that there's a phenotype that's actually quite complex, and we'll see electron microscopy images of this in a little bit. This is important for digitization because it gives us landmarks to use for different techniques. So these are two examples of the Basilaria diatom. So this is a bunch of these rod-like cells. They're in physical contact in a colony, and they tend to slide against one another. If they're quiescent, of course, they sit like this, as you can see, in this sort of stack. Sometimes that's elongated, sometimes it's sort of in a short stack that is just one cell next to another, but sometimes they slide against one another, and they make this accordion-like movement. So these are some examples of diatoms in the wild. You can see it's very hard to see them in the culture here that we have under the microscope, but you can see the movement here in this movie. So this movie will show one oscillation. So the cells unfold. They unfold as they slide against one another, and then they come together, they slide back against one another to form this accordion-like movement that oscillates over time. As you can see, they're sliding back against one another, and then they reach a maximum limit, so they don't quite come apart, and then they come back and they slide back around. This becomes important later in some of our measurement techniques because we do get this oscillation, first of all, that where they're fully extended and then they slide back together and then they come back and extend out in the other direction. But also because there are these physical limits to the colony's extent. So as they slide apart, they don't slide apart altogether. And this is going out of focus a little bit. They kind of have a place where they stop, and there's limited contact at that point, and then they come back together. And there's some physics of movement during the end of that slide that are interesting to characterize. So this is a clearer view of one colony that's isolated from the rest of them. So you can see here it's a much cleaner view of the movement. So it slides apart, and the cells are almost like coming out of contact. There's a small area where they remain in contact at the very end of that extension. And then they slide back in the other direction, and you can see they don't quite come out of contact. They stay in contact a little bit at the ends of the cell. And so they do this sliding motion And this is a characteristic movement of the Basilaria genus. And so this is what we're going to be characterizing as well as the phenotype, this movement. So we can analyze Basilaria movement in culture. This is an accordion-like movement. We can model this as a series of oscillators. So as you can see here, as we look at this oscillation, we can actually look at this as oscillators, both cell-by-cell oscillators, or sometimes we can measure movement relative to different cells in the culture. So there's a leading cell, as you can see, there's a leading edge of the movement, and then there's a trailing edge of the movement. So the one cell in the middle of the frame isn't moving at all. The rest of the cells are moving back and forth. And you can see the cell at the top, or the cell sort of to the farthest right, is the leading edge of this. So it's really driving the oscillation. The rest of the cells, except for the bottom cell in the stack, are following, and it's producing this sliding motion. So you'll see that there are different phases of movement. There are different ways we can model this. We can model this as an oscillator, but to measure this in sort of a collective motion movement, there are a lot of interesting techniques we can use. So we can also model. Okay. So we can also model this as a series, a couple of oscillators, and this was an idealized version of it. But we also have, this looks a little bit more like what you would see in the wild. So that pure oscillation isn't the only behavior that's going on. Sometimes the movement can vary due to stimuli. And we're not varying the stimuli here, but sometimes these colonies will become quiescent, or they'll change their oscillation, the rate of oscillation based on the amount of light that they encounter, based on their chemical environment, and so on. And so you can see here, this is a little bit more natural set of oscillations, in that you have a little bit more variety. So you can see that there's variation in the colony. It's not just that smooth oscillation of a couple cells. You have stacks that are many cells wide, and they move in different ways. And so some of this is stimulus driven, some of this is driven by other things. And we'll talk about some of the candidates for why this motion is occurring later. So here's some electron microscope images of Basilaria colonies. So at the top, you can see one of these stacks, and they're arrayed kind of a semi-open array here. So there's a leading edge that's kind of moving out, and there's a trailing edge that's sitting stationary. And you can see how it's kind of spreading out like an accordion. But you can actually zoom in on single cells and look at some other features. So like we talked about in the first slide, we have this raphe, which we can use as a landmark. We have other things along the surface of the cell we can use as a landmark. And depending on the resolution of the input images we're using, we can actually characterize some of these things and use them as landmarks for marking certain parts of the cell relative to other cells. And so this is a very, very high resolution image. This is 1 micron, this line, this is 2 microns, so you can see the scale, the spatial scale of this. The entire cell is about 10 microns, so that's a very, very tiny cell. And you can see how small these features are. These feature sizes are, in many cases, less than a micron. So these parts of the raphe here are, maybe 2 microns at most. And some of these perforations are, a fraction of a micron. So very small feature sizes. So one of the goals of the digital Basilaria is to characterize the cells, to find landmarks on the cells, and to find a characterization of the movement and collective behavior of these colonies. So one set of experiments we did was to apply deep learning and do a deep learning analysis to these Basilaria colonies. And a lot of the technical work here was done by Ujjwal Singh and Asmit Singh, who have been contributors to the DivaWorm group, especially back in 2018, 2019, 2020, in that era. And so we can use deep learning techniques to define individual cells in a Basilaria colony from movies of isolated colonies. And you saw the movies previously. We can isolate the colonies, and those are the best sources of data we have to look at these colonial behaviors, but we can also use videos that you saw where you have multiple colonies, you have some diversity. But in this case, we just use some isolated colonies, some very simple movements, smaller colonies, just so we can get a handle on the sort of structure of these colonies, the structure of the cells, and place them in a spatial reference frame. But the cells themselves, you can see that we can also characterize different parts of the cell. This becomes important for movement because remember I said that these cells slide against one another, but they don't come out of contact. There's always a little bit of the cell that remains in contact, even at their maximum extent of movement. So you see that we've defined a number of landmarks on each cell. So we've defined 3 landmarks where you have each end and then the centroid. So the centroid is just the midpoint between the 2 ends. And we can identify those through computer vision techniques. And then we can define a bounding box defined by 1, 2, 3, 4, and 5. 4 and 5 being each side of the cell. So basically we're creating a bounding box around this elongated cell. We're defining the ends of the cell. We're defining the left and right edges of the cell, and then the centroid, which helps us to sort of register all of that. So from that, we can do this for each cell, and we can derive positional coordinates. So we can go across images of different colonies and build a spatial reference frame with positional coordinates and place these in a positional coordinate system. And this allows us to build graphs from the data to sort of characterize the shape of these colonies in terms of numbers, but also to create higher order shape parameters. So we'll talk about that in a little bit, but basically to define the shape of these colonies as they're moving. To do this work, we used DeepLab version 3, and we used our Open DevoCell platform, which is a platform internal to our group, with some mask preprocessing. So this is a little bit older work. It's not the cutting-edge state-of-the-art work that you might expect, but again, it was a pretty decent result. So if people are interested in participating in this and doing another version of this, we'd be happy to talk to you about that. So this is what the result looks like after segmentation, where we have these bounding boxes that represent these cells. So you can see it's not perfect by any means, but it does give you nice bounding boxes that approximate the cells in movement, which is critical to what we're trying to do here. So here's an example of how we put this into a spatial reference frame. This is this colony. It's expanding. It's in movement at a certain rate. You can see it's not a clean movement. You have some. You actually have a little bit of a. It's not directly an accordion-like thing. It looks like it's kind of like moving in 2 different directions. So it's not really clear what the input data is. Sometimes these input data are very messy and hard to really see with the naked eye, and so that's why we are applying, one reason we're applying computer vision to this. But in any case, we can get a fixation on the centroid of each cell. We can plot that centroid value here in an XY coordinate system. So we can tell you where the centroids of the cells are, and you can see that sometimes the centroids are close together and sometimes they're far apart, and that approximates the movement across this colony, or the position across this colony, and differences in movement. So the differences in these centroid positions, if we take the first derivative of position, we say that that's some sort of motion in that direction. So if we take different images from different time points, we can plot these out and see the motion or the changes in position over time. So we can look at another example where we can see the same thing here. We have this cell, and we have to orient these cells in different ways so we can turn the orientation around. We tried to orient them in at the same orientation, so there was some pre-processing done there. But basically, we have the same situation here, where we have the centroids that are far apart and close together, and the close together ones are when the colony is folded together, and the ones that are farther apart indicate there's some sort of movement there, extension of movement across the colony. And so this is a third example where we have much more regular spacing between the centroids, and this is when the colony is expanded in this manner. So in this case, there are 2 leading edges, cells that are 2 leading edges, and they're kind of, there's a center of mass up here where it's closed. And so you can see that movement, but they're much more evenly spaced, centroids are. And so you can see that it's reflected, what you see in the image here is reflected on the graph. And then a final example is where we have this, which is a similar type of configuration, but it's flipped in another direction. So, we can do things like, we can rectify these signals or put them in, transform them into another coordinate space or run a PCA analysis on these. These are raw positions, so we can actually do a lot of things with them once we get these into a bivariate context. So one of our other group members, Dr. Richard Gordon, he published a book chapter in 2021. It's called The Whimsical History of Proposed Motors for Diatom Motility. And this is chapter 14 in the book, Diatom Gliding Motility. So this chapter focused on the different mechanisms that have been proposed for diatom movement. As I said before, we know that they move. We know that they have these complex movements, but we know they also don't have a brain and they don't have muscles. They have actin filaments, but they don't have muscles. And so what is driving this motion? What does it sort of resemble? What is it approximating? And so people, and this goes back to like the 17th century or maybe the 18th century, where people have been proposing different types of mechanisms for movement. And so they've used these sort of analogies. They've used other organisms like snails and humans. They've used human activities like, rowing on a kayak. They've used very dated references. Like there's one example where there are a bunch of people rowing a rowboat, and you have another example of like a jet engine. And, there's another example of a pinion on a rack. So there are a lot of ways that people would propose that movement occurs. And some of these are very specific to the time period. So like the jet engine or the rack and pinion. Others are sort of these analogies to human work with our muscles, with machines or cultural artifacts. And then some still are other organisms that have actual muscles that are, doing work. And so we're, just moving around. And so there are a lot of different potential mechanisms, or I guess you could call them metaphors for movement. In this book chapter, Dr. Gordon talks about 21 different mechanisms that have been proposed for movement. But we're still no closer really to understanding movement. It's actually a pretty complex process for these tiny organisms that don't have a brain, they don't have muscles, they have actin filaments, and they can behave in these colonies. So what exactly are the mechanisms? And we'll be going over that next. So to get at this question, we actually wanted to model the cells in the colonies a little bit differently. So Thomas Harbeck, who is an independent researcher in Germany, has harvested diatoms from the Neckar River in Germany. And he's used his home microscope to look at these diatoms and produce these movies that you see here. So these are not too bad, they look like they're pretty good, ways to experiment with these colonies to sort of break down individual cells and the relationship between the cells. The movies that we had in the deep learning experiments were collected from the web, so people, amateur diatom observers have taken movies of this, and some of them are not of high quality. These movies are much higher quality. So Thomas, as a retired physicist, he actually has access to some of these physics tools, these biomechanical tools, and biomechanical models to analyze the data. So this is from our contribution to the Diatom Gliding Motility book, chapter 10. This is from Towards a Digital Diatom. And this shows how we did this modeling. So each diatom cell is independent, and you measure the distance between, if we take the extent of motion outward, we can take the distance between the neighboring cell, the end of the neighboring cell, and the end of the cell that we're measuring. So diatom number 1, for example, there's a 57.5 micron distance between the far end of diatom number 1 and the far end of diatom number 2. Then we can do the same for diatom number 2 relative to diatom number 3, and we get a little bit different measurement. It's a little bit larger, 59.14 microns. And we can do this all the way down the colony. So we're not using landmarks like we were in the last paper. We're in the last set of experiments. We're just using these landmarks and measuring the distance between the landmarks. We don't even really need to define the landmarks too carefully. We just need to find the extent of the extension of each cell along this colony. So we're able to measure that, and we're able to use that to track each cell as it's moving across this accordion-like structure. And so we can actually look at the distance, which changes as the colony contracts or expands. And so this is the farthest extent of the colony. These distances will get smaller as the colony comes back together, and eventually will be 0 when the colony is fully contracted. And then when it goes in the other direction, these numbers will get bigger. We can also do really interesting things that are, have their home in statistical physics. And so one thing we can do is look at a phase portrait of diatom oscillations. So when we do these measurements, we can actually get these, record like a measurement that goes from, say, 57.5 microns down to 0 microns, and then 57 microns in the other direction. So from that, we can look at 2 neighboring cells, in this case, diatom number 1, diatom number 2, and draw phase portraits that describe the motion relative to one another. So as you can see, we have a signal here that goes back and forth between one side and one extent and then the other extent as you go in the other direction. And you can see this phase portrait. And so you notice that the phase portrait is nice and round, nice and circular, it's a nice orbit, but you do have these points here, which are squiggly, as opposed to the rest of the phase portrait. Actually, you see some squiggle in it. There's variation in the movement, and the reasons for that, which are partially anatomical and partially because of the medium that the diatoms are in. But you have this sort of area where it really gets squiggly. And what these are is when you get to that extent of the motion, this largest extent of the colony, you end up with this end of the oscillation. So the oscillation extends to it extends all the way out, and then it stops and moves in the other direction. When it stops and moves in the other direction, this is a signature of, we expect that there's some sort of signature of jerkiness, which is something like the fourth derivative of position. So we have these, we can calculate position at any one point. When we put that together, we get a phase portrait. And then as that changes, we get acceleration. So we get acceleration that we can measure here. We can integrate over a certain number of time points and get acceleration. And then at the fourth derivative of position, we have this jerkiness, which is very quick. It's not acceleration. It's actually something more higher order than that. So actually, it's a third derivative of position. I'm sorry. So this is something that we can look at. We need to apply better tools to this. But in this paper, we just basically suggested that this was what was going on here. And so, you see there's a lot of things you can extract from these phase portraits. And you can also plot them against sine waves. So we can have a sine wave at a certain frequency. We can look at these oscillations as something that occurs over a certain frequency. So you can see that in the full sort of extent when the thing is oscillating like an accordion, this is going, it's pretty much consistent with the sine wave. Sometimes you get shorter get shorter changes in position here, so it doesn't track exactly with the sine wave. But this could be like changes in direction. This could be just a slowing down. There are a lot of things that could explain this. If we look at this mapping to a sine wave more specifically, we can see that you have these variations in the amplitude, but not in the phase, and so that's good. That suggests it's basically a sine wave, and we can actually look at different. Maybe we could take this a little bit further and say, if we expose the diatom to different types of stimulus, or you put them in different media, what does this look like versus the sine waves? The sine wave is like a null hypothesis or just a random model. So you can actually compare it against a sine wave. What's interesting as well is you can watch them changes in behavioral phase. So, these uh, diatoms are oscillating back and forth. We can compare that to a sine wave. Sometimes there are changes in the amplitude of the signal, but sometimes the signal ends or it attenuates down to zero. And what this means is that the diatom is starting to stop oscillating. Basically, it's oscillating to swim and it's becoming quiescent. So you can actually observe this in the data where it becomes quiescent. And so this is where an oscillation slows to quiescence. We can observe that. We don't see a lot of noise associated with that necessarily, but then again, we've not done a huge number of observations of this. So it would be an interesting question for future research. This actually can be applied to other types of things as well. So there are these things in physics that people, or biophysics people, are exploring called microswimmers. And so, like I said, the oscillation and the patterns of oscillation have to do with swimming behaviors for these colonies. They're swimming through a water column, and the water sometimes changes its composition. Sometimes it's, salinity, its brackishness, etc. Sometimes it's exposure to light, and it's swimming through the water column. It's trying to propel itself. And so the lessons we learn from the Basilaria and even other diatoms for that matter can be applied to microswimmers. So microswimmers, this is an up-and-coming area of biophysics. There are different things you can explore here. This is a magnetic helical microswimmer. So this is a magnetic microswimmer that's made of a metal, and then it's put in a magnetic field, and it can move helically. So it's swimming helically. There are no muscles, there's no actin, there's no brain, but it's responding to a magnetic field, and it's moving accordingly through a medium. On the left, we have thermoelectric guidance. So we have these little microswimmers, which again are about the size of a diatom, and they move around, these little balls, and they move through this water column or this liquid column, and this is what they call thermoelectric guidance. So you can use this to guide microswimmers to different targets as well. Microswimmers are very useful for different things at the nanoscale or the microscale, things like their medical applications, their other types of nanotechnology applications. So we can learn a lot of lessons from the diatoms and from Basilaria specifically to apply to things like this. Now that we've talked about some of the work of characterizing the phenotype of diatoms, in particular Basilaria, the second part of this talk will get into the sensory world, or the habitats experienced by diatoms. And so you can see that diatoms they exist in these tidal or coastal ecosystems. So you have estuaries, which you see in the upper left. You have these tidal areas where they're near the surface of the water or near like the shoreline, the river, there are a lot of rivers and streams where they live. So some of the specimens that Thomas Harbeck got were from the Neckar River in Germany. So these are temperate streams and rivers and lakes that harbor diatoms. You can also harvest them from estuaries or from anything near the surface of the water column. And so you can go in and you can get a jar of water and you can isolate these diatoms. If you go into any one of these environments, you should be able to find them, isolate them, and culture them on your own. They're difficult to culture, but not as difficult as other cell lines and organisms. So they're probably about as difficult to isolate and culture as C. elegans, which is, to say that you need to have some equipment for it, but not too much. You can easily, they're plentiful in the environment. So this is the kind of sensory world that they're used to. They're used to light. They're used to changes in the light column because water, once light from the environment hits the water, there's a degrading, it's refracted, of course, through the water column, but it also degrade, it doesn't penetrate the water column that deeply. So by the time you get to maybe 15 or 20 meters, it gets pretty dark. And you can see that in this picture at the bottom. So one thing we're interested in is their responses to light, their responses to chemical variation. In a lot of these brackish environments, you have salt water and fresh water, so you have this mix of things. And so there's a mix of salinity gradients that are kind of interesting. There's also, differences in light, differences in water temperature, and other things. So there's all sorts of, stimulus changes that you can observe and that these diatoms have a response to. So one interesting area of research that kind of is in the same vein as this is psychophysics. And so psychophysics has been applied to a number of animal models. This reference by Stebbins from 1970 talks about animal psychophysics. So designing experiments that are optimized for animals. They mean non-human animals, but of course you can apply psychophysics to humans. You can also apply psychophysics to mouse. So this is where mice are looking at cats and other mice. And then you have goldfish, and then maybe even diatoms. Now, no one's done work in diatoms in terms of psychophysics, but that's kind of what we're proposing happen. And so Saris published something in 2006 on the relational psychophysics in humans and animals. So there are a lot of psychophysical techniques that are quantitative that lend themselves to computational analysis or computational characterization, and they really relate to how these organisms respond to their environment and other, physical properties of the environment. So one of course, the big objections here as well, like I said, diatoms don't have a brain. So how do we account for that? Because those other organisms had a brain. And so what are we even talking about? Why do we care about psychophysics? So there are a number of papers that discuss the idea of non-neuronal cognition. And so we can think of cognition as this information processing mechanism, or this information processing capacity that doesn't necessarily rely on a brain. It relies on complexity, biological complexity, and it relies on different mechanisms for information processing. So in this paper by Lyon in 2015, this is the cognitive cell, bacterial behavior reconsidered. They consider bacterial behavior, and bacteria also do not have a brain, as non-neuronal cognition. And they define non-neuronal cognition as the total set of mechanisms and processes that underlie information acquisition, storage, processing, utilization. So we have information acquisition, information storage, information processing, information utilization. It doesn't require brain. It doesn't require a brain. It doesn't require neurons. It requires certain, features in both the individual cell and in a colony to do this. And so we're going to see some of those mechanisms later. Beluska and Levin talk about the extracellular matrix being ideal as a memory medium. So they talk about the extracellular matrix being important as memory, and there being an internal model in the cell that interacts with the intercellular matrix that produces stigmergy, which are the physical traces that can be referenced and read in the future. So if you're familiar with insect colonies, like ant colonies or termite mounds, they use pheromones, and they lay pheromone trails down in their environment. In other conspecifics, other insects can read those signals and follow those trails, and they have an internal model inside of their physiology that tells them to recognize that and to follow that trail. So these are all things that we can use in diatoms as well. So in diatoms, there's an interesting extracellular sort of stigmergic mechanism, and then there's these mucilage trails. So these mucilage trails are secreted polymers that completely encase the cell and are responsible for adhesion and interactions at the internal environment. So these mucilage trails and strands, they work to connect these cells together. So the cells, when you looked at those movies of the motion, they appeared to be sticky, so they didn't fly apart when they extended themselves out, but they also didn't move smoothly. There was some noise, and that noise was because you have these mucilage strands and trails in between the cells, but also they're laid out in the environment so that the other cells know where to go. They have a signal in the environment. They're responsible for a number of interactions with the external environment. So we can actually also look, though, at the way in which diatoms process environmental signals. And so, like I said, there's a lot of variation in the environment they actually do take in environmental signals like light and chemistry and other things. And so what are there's a pretty rich literature in the diatom community of some of these sensory um, interactions. So this first paper is the effects of elevated CO2 on the natural diatom community in the subtropical Northeast Atlantic. So this isn't just a standard diatom community. There isn't a lot of work done specifically in bacillary, but the idea is to characterize its been in diatoms and then apply it to bacillary. Bacillary may vary in the way this is done there, but it gives us a model to go forward. So the diatom cells can sense CO2 sensation and concentration, and that affects growth of the cells and growth of the colony. In cases of high CO2, there's reduced grazing pressure and an effect on photosynthesis. And then in this paper, Hayden, light intensity modulates the response of 2 Antarctic diatom species to ocean acidification. There are 2 things that we see. The first is that CO2 and light availability affect diatom physiology. They tested 3 light intensities, and they saw that low light affects physiology and distinct physiological traits. They also observe localized sources of chemokinetic and chemotactic mobility. So chemokinetic is where you have chemical signals that drive movement, and then chemotactic is where chemical signals drive movement in a certain direction. So there are 2 different things that they're observing here. They actually observed attraction, pheromone-guided search to find mates. So this is interesting because there's this pheromone-guided search to find mates and to restore cell size. And diatoms have this weird reproductive capacity. Basically, they accrete material to restore their cell size. They also do mating in some way. So there's this relationship between these chemical sensation, movement, and even other types of behaviors. This paper by Bondoc, Decision-Making Physiology of the Benthic Diatom, Seminavis robusta, Searching for Inorganic Nutrients and Pheromones, and this other paper by Falciatore, Perception of Environmental Signals by a Marine Diatom. This talks about diatoms having sensory systems for detecting and responding to fluid motion, or specifically shear, osmotic stresses, and iron abundance. So they have these senses for chemical imbalances in the environment, but also physical forces in the environment. So this is important when you're in a water column. The nutrients are fluctuating, but also you're in different parts of the water column. There's different shear forces happening, and so it guides your motility. So now we're interested in building a model around propulsion as a mode of movement. So we want to build a psychophysics model that focuses on propulsion. It focuses on maybe having like sensory inputs or other types of inputs that result in motion and variation in motion. So we're interested in environmental inputs and actin filament outputs, actin filaments being the things that drive motion. We have oscillatory movements as a small subset of behavior, but this allows us to view what we'll call the COPG, or collective pattern generation circuit. We'll talk about what that is in a minute. And then psychophysics models account for a wide range of behaviors outside of oscillatory movements. So we can actually look at other types of behaviors outside of this, but we're just going to focus on the oscillatory movements for now to sort of propose a set of psychophysics models for this work. So this is movement generation. This is the raphe wall, and there's a substratum. So if you put the cell on a substratum with the wall on the outside, of course, it can move against that wall. The substrate could be water, it could be another cell, so another raphe wall. You can vary the substratum, and it gives you, different results. But basically, it's moving across the substrate in one direction, and this is gliding locomotion. So they're actually looking at the mechanics of this in this paper. This is an example of how this works. So there's a cell wall, substrate, and the movement. As we can see, as it's moving, there are a lot of interactions here. We have the actin filaments inside the cell. We have this myosin, which is moving against the actin filaments. We have a membrane complex at the surface of the raphe wall that transmit the forces downward to these mucilage adhesives, and then those mucilage adhesives transmit those forces against the substrate. So you have this interaction between the raphe wall and the substrate through these adhesive mucilages. They're transmitting forces from inside the cell, and that's how you get this movement. But also you get collective movements when two cells are moving against one another, when the substrate is another cell raphe. We can use a series of coupled sinusoids to model an ideal collective pattern generator. So again, we'll talk about these collective pattern generators, and we'll recall those sine waves that we modeled for basic bacillary movement, and we'll consider that for some of our collective pattern generation work. So we have these half phase or 90 degrees out of phase movements. We have quarter phase or 45 degrees out of phase movements, and those can be used to simulate discoordination. So in the cases where, maybe the colony breaks apart, or it's not moving in the way that maybe we expect it to given the stimulus, or when we change the stimulus, it becomes discoordinated. That's what we'll be doing. So we can see examples down below where we have these out of phase sinusoids. So we'll model each cell as a perfect sinusoid and then vary that outward. So we can change the phase even to something anti-phase if necessary. Additive noise can also be used to simulate discoordination. So in the graph on the right, we can see additive noise. This is kind of an extreme version of additive noise, but we have this, we can add this in, and this is what it looks like. So we can actually have two, half phase or quarter phase signals with noise, and that can be used to model movement as well. And then finally, the speed of movement or the derivative's position can also be varied according to the effects of the stimulus. So here we have an example of a central pattern generator. This is something we find in nervous systems. This is a series of neurons that are interacting. You have these excitatory synapses and inhibitory synapses that are driving this oscillation. So basically you have neurons that are signaling each other in an excitatory or inhibitory fashion, and they're driving this oscillation in the circuit. So you can see that there's an input from C1 and C2, and their feedbacks between X1 and X2. X1 and X2 are both being modulated by C1 and C2, as well as being co-modulating each other and responding to external inputs. So you can see how that works. That's a collective, or that's a central pattern generator, and we see this in like the spinal cord. We see this in other parts of like insect nervous systems. We see this in mammalian nervous systems where you need to generate a rhythm for something. So you see this in the cardiac system and other places. It generates a rhythm that's continuous, so it's just the activation of this circuit, and it generates a rhythm that's a central pattern. So this is a central pattern generator. It's been talked about in the neuroscience literature for decades. What we're talking about here is a collective pattern generator, which is different. In a collective pattern generator, we have these inputs to the leading edge cell, as we saw in our movies and we discussed already. So this leading edge cell will take in these sensory inputs. And then we have these, they're landmarks. They're just sites where you have sensory inputs. And then those sensory inputs are linked to other cells down the colony. So you have these links, these mechanical links. You have other sensory links between cells. And so that's what this network is. So it's just a collection of sites where either you have an input of sensory information or you have an interaction with another cell. And so we use the W subscript for these to identify these different sites. And then we have a network of different relationships between these points. So we can see that because these cells move against one another and they're all experiencing the environment in different spatial locations, you have a lot of potential interactions between them. And so that's what these lines are, they're interactions. it's almost kind of like a neural network, but it's not quite because it's structured. First of all, it's spatially inhomogeneous, but also because it's actually more of a interaction model than a neural network. So we can model this and we can look at the variation in these different interactions, the strength of the interaction over movement, over stimulus intensity, and over time. So our sensory inputs are I sub n. These can be varied. I just put 3 here as an example. These are sensory inputs at inertial points on a surface of the leading cell. Then we have movement weights. So the movement weights are these weights of interaction. These are, the way we've modeled this is largely about movement and about the interactions between raphe walls, but we can also model some of these W sub MN points as environmental inputs as well, depending on the movement of the colony, depending on which ones are exposed to the environment at different times. And then, of course, there are also interaction points for mechanics and movement. So this is what we're talking about. And so the idea is that this model, if we modeled it, should describe these interactions and should describe the movement. So using our COPG model, we can look at differences in stretch phase between coupled cells. So on the left, we have this model, this idealized model of our COPG. And then we have directional movement back and forth. And then we have microscopy images where we have an example from these images. So we can see that we've kind of labeled the leading edge. In this case, I think we've labeled the trailing edge here, but we're kind of pretending that that's the leading edge of the cell. So wherever, well, you could have environmental inputs on the trailing edge as well. But in any case, we have one cell where these inputs are coming in, and of course, ideally, they could come into any cell. And then we have movement back and forth. So you can see that these inputs, these environmental inputs, are transmitted across the colony as it moves back and forth. And so based on the configuration, you can see the spatial extent of the oscillation, these inputs on a single cell, and then these networks being arbitrary points within this colony. So there's a network that forms within the colony that's a network of information processing. And so that's basically the idea. And then as you vary the environmental signals, this network changes in terms of information processing. It drives the oscillation, which is not autonomous movement, but it affects that in different ways. We can also look then, when we're talking about environmental inputs, at sensory thresholds or just noticeable differences. So this is another core attribute of psychophysics. This describes differences in light intensity, for example, measured by output movement weights. So what this means is that you have a light source. And again, I have the sun there, but you're going to probably have it attenuated by the water column. These light sources are going to come into the different points at different intensities. And we expect that there are these sensory thresholds where the light is intense enough to trigger a change. Sometimes light will be, normal, or it will be, not noticeably different from normal. So it might fluctuate a little bit, but it might not affect the movement that much. But sometimes it's going to be above that noticeable difference threshold and affect the information processing network. And so we can measure those differences in light intensity by looking at the structure of this network, looking at the motion, and even modeling the diatom and proposing that there are different, noticeable difference thresholds or other types of information processing properties of this network. We can also look at the signal to noise ratio. So one of the reasons I put this noisy signal in is to point out that there is a lot of noise in the environment. And so, the ability of the diatom to separate out that signal from the noise, especially in terms of measuring forces or measuring chemical inputs, is important. So there's this dynamic sensing of the water column, the physical properties measured by output movement weights. So when there's an input, again, there's a lot of noise to that input. Can this information processing networks separate the signal from the noise and operate on the signal? And that's another thing we can think about in terms of psychophysics, because signal to noise ratio is something we can play around with and see if it makes an effect and makes a difference on the movement. I should note that when we looked at those phase portraits of the movement between two cells, that we saw these noisy fluctuations in those orbits, and some of that is due to sort of the extent of the colony moving against to its largest extent spatially. But the other part of that is noise inherent in this movement against one of the cells moving against one another, and perhaps some of that is due to this information processing within the colony. And so that's something we can look at more deeply as well using this kind of method. So one example of psychophysics is Weber's law. So this is where we look at the intensity of the light stimulus versus the ability to discriminate different light sources. So sometimes the light isn't coming from one direction, as you saw in these environments. You have light is being diffracted, and so it's coming from different directions. There's this sort of effect when you're underwater. That is where the light is indirect. It's kind of I can't remember the name of the effect, but it diffuses the light source enough so that the diatom needs to be able to discriminate between the true light source or different light sources, and this holds true with chemical inputs and physical inputs as well, especially chemical inputs when you have a lot of different chemical compositions in the environment that need to be discriminated against. So we can use something like Weber's law to quantify these more directly and do experiments like that. So I've talked about a lot of things in this talk. This is our roadmap for the digital bacillary. So far I've talked about modeling, I've talked about data segmentation, extraction, I've talked about characterization. So we have this roadmap for the digital bacillary. We're operating mostly from microscopy data, but some of that also involves, behavioral data and maybe even simulating conditions. But let's take the microscopy data as the root for our roadmap. What we do need a lot of microscopy data on movement, on movies, data on static observations, and so forth. And those lead down two different paths. We have a deep learning path and a physical modeling path. And the physical modeling path is heavily dependent on dynamical systems, and the deep learning path allows us to look at things like colony shape. And within colony shape, we have a number of things that we would like to look at. We'd like to get accounts of the positions that these cells take with relation to a colony. We have variation of that shape or of the colony shape. So we saw those images where you have a number of different colonies in the same image, and they all look different. They all had different compositions, and they all had different properties. And as well, we also want to look at things like topological properties of the colony and its shape. So using a topological data analysis approach that's with respect to time. So we can use deep learning for that. We can also use the psychophysical modeling for that part too. In the terms of the physical modeling, we are directly addressing dynamical systems. So there are a number of things that are interesting in dynamical systems land. Some of those are like the collective pattern generators, the psychophysics themselves, and the moments of position. And you saw examples of phase diagrams and other work that, we have information processing networks and, other types of things that are very interesting. So that's kind of our roadmap for developing the digital bacillary. And hopefully, in doing this, we build a better sort of account of diatoms, of bacillary. And why do we care about diatoms? Well, diatoms are very important first of all, they represent a lot of biomass on Earth. They're also applicable to things like microswimmers or nanotechnology. And, they're also, they're important in like biofuel production and other things. So understanding how we can culture diatoms, how we can, address their behavioral variability and their physical variability is important. So thank you for your attention. This work was done within the DivaWorm group. So you can visit our website, our GitHub repository. And we also have a deep learning platform called DivaLearn, which involves graph neural networks and pre-trained deep learning models and other types of work, and that can be found at that repository. Um, Thomas Harbeck has run a website called Observation of Diatoms, where he's put out a lot of his observations on culturing diatoms and taking images of them. And then check out our weekly Devo group meetings. Sometimes we talk about the digital diatom, and sometimes we talk about a lot of other really interesting topics in computational biology, developmental biology, and biophysics. So thank you for your attention.